The webinar has now started.
Hi, everybody. We're going to wait and make sure we get everybody in. We'll wait just a couple minutes and then we will start this wonderful webinar.
I think there might be some confusion on the links because I think that there's some of our staff are trying to get in and are not able to. You were, and then what happened?
My suggestion is that at 12:05 we begin because it will be recorded and we have about 26 people on.
Katrina, are you letting people in from the waiting room? Because our staff is in the waiting room.
Yes, everything has begun. And we have
Are you still in the waiting room?
28 attendees.
I don't see you.
Stephanie, are you prepared to go ahead and welcome and introduce our webinar today?
I am. So hi, everybody. This is Stephanie with It's the Journey. So happy that you guys are joining us and so happy that we're able to put this this wonderful webinar on. Nancy Paris and I from Georgia Core wanted to kind of give everybody an update on where your dollars have been going. I know we talk a lot about where the dollars are going for our grantees for mammograms and for genetic testing, but these are researchers that your dollars have funded, and we really wanted to get an update. Of course, we would love to be in person, but we really wanted to kind of get an update on the progress that they've made and the things that they've been able to do with the dollars that you guys are walking 30 miles every year to do. So Nancy at Georgia Core, we've had a relationship for 3 or 4 years, I think, right? Even before I got here. So that relationship had started when we kind of wanted to gear towards research, of course. And all of these researchers are here in the lovely state of Georgia, even though I think it's raining today, kind of raining today. But they're all here in Georgia. I haven't been here long enough to love Georgia probably as much as they do, but I am a Falcons fan, okay, and a Braves fan. So without further ado, Nancy, I'm going to kind of pass it off to you, and I'm really looking forward to hearing what everyone's doing. So thanks for joining us, and I'm going to let Nancy take it away.
Thank you, Stephanie, and all our partners at It's the Journey. I'm Nancy Paris with Georgia Core, and I'm excited to be the moderator. This is the first such webinar we've tried with 4 innovative, dynamic researchers you're going to hear from in just a few moments. I want to thank all of you for your support of cancer research, and we want to make sure that you have the opportunity to hear about the research that's going on in Georgia and understand a little bit more about the importance of it. I think you've seen the pictures on the flyer of this team. They're diverse and their bios are absolutely outstanding. So our panelists today are, they're all PhDs, so I'll just say Dr. Ritu Aneja, who is a professor and the director of the graduate program in biology at Georgia State University. Dr. Cheryl Gomillion, who is assistant professor of chemical materials and engineering at the University of Georgia. Dr. Adam Marcus, who is professor at Emory University in the Department of Hematology and Oncology, and also the associate director for basic research. And Dr. Mandi Murph, associate professor of pharmaceutical and biomedical sciences at the University of Georgia. So I'm sure that you can see from their backgrounds, they're highly qualified, but also bringing engineering, pharmaceuticals, biology, hematology, and oncology together, you'll get a real good feel of the depth of research. We've just asked each one of them to talk for about 3 or 4 minutes on this theme of highlights and hopes and research. They'll tell you about their research, and they'll also say something personal about how they got interested in breast cancer research and what compels them to devote their lives to this extraordinary work. So thank you again for this opportunity to collaborate. We want to reinforce that this makes it possible for It's the Journey and for Georgia Core to provide more funding together to these great teams of researchers. And it also eliminates the opportunity for them to write multiple grants. And I think they appreciate that. In fact, I've heard that they do. So there are lots of benefits to it. And if you like this program at the end, we hope you'll let us know, because there are other teams of researchers with whom you may want, from whom you may want to hear, and we'd like to share their work with you as well. So having said all that, what we will do, so Pam, do you have, if you want to show the slide for each researcher, just in the order that you have them, just the first one, then I can call on that person and ask them to begin. Are you there? Or I can get started either way.
Oh, there we go.
Okay, screen sharing. This is so cool. Okay, we're going to go to Adam Marcus. So Adam, would you just give us a background of your research focus and your personal interest in research? And if you see me waving a hand, it means that you've gone more than 4 minutes.
Sure, that's a good thing, because I'm known to carry on. well, thank you for having me here, and thank you everybody for spending this time with us. I also want to say thank you to It's the Journey and Georgia Core for their support over these years. I mean, it's really been instrumental to the breast cancer research that we've done. So I'll be brief. My laboratory studies how breast cancer metastasizes, how it spreads. So we're fascinated by looking at specific individual and rare cancer cells and actually being able to pull out these rare cancer cells that we know are drivers for cancer spread, that we know are drivers for recurrence, and really for the first time, pull out these cancer cells and look at how they work. So we're very fascinated in understanding how these cancer cells work, because in our opinion, really the only way to continue to develop effective treatments for breast cancer is to understand the sort of underpinnings of their behavior. So we have a technology that we developed that specifically goes in and isolates single cancer cells. And this funding has allowed us to develop this technology even further. And for the first time, we're able to pluck out essentially single breast cancer cells, look at how they work. And then our ultimate goal now is to develop treatments for these specific cancer cells that cause recurrence, that cause spread. And that's ultimately what my laboratory is focused on, is how do we stop cancer spread, and that's what I'm passionate about. That's what sort of drives me, is to understand how we could develop these new treatments and help people. I'll stop there. And yeah, thanks for listening, everyone.
Perfect. So Pam, you want to put up the introduction for our next presenter, please? This is Dr. Mandi Murph. Dr. Murph.
Hi, can you see me?
Hi. I can't see you. I see, there you are. Welcome. So please, same thing, give us an overview of your research and a little bit about you personally.
Okay, so I did my undergraduate degree at Emory University in Atlanta. I also received my PhD from the Georgia Institute of Technology, where I got really interested in disease mechanisms of cancer. So I did a postdoctoral fellowship at the MD Anderson Cancer Center in Houston, and it's there that I got really passionate about cancer research. And it's hard not to feel that way when you're in and out of the hospital, surrounded by so many patients, the breast oncology unit, and so forth. It's just really impactful. And so I work at the University of Georgia, and I'm in the College of Pharmacy. And what that means is that my research has to be related to drugs and drug targeting, perhaps drug kinetics, but something that is pharmacy related. But I'm a biologist by training. And so my research is focused on, you might say, nanoparticle delivery. What's inside the nanoparticle? How do we get it to specific places and specific sites within the body? And we're very interested in answering the question of how can we drug target triple negative breast cancer? Previously, until about a year ago, there were no specific drugs for triple negative breast cancer. They don't express the estrogen receptor, the progesterone receptor, the HER2 receptor. There were not targets on the outside of those breast cancer cells. And so we basically wanted to drug target perhaps the undruggable. But again, that's changed in the last year or so, where we now have a couple of FDA approved drugs for it. So that's fantastic. And so we try to assess that question. How can we target it? And so again, we're doing that with nanoparticles, and we're looking at inserting different unique kind of entities into those particles to reach the site. My interest in breast cancer started at MD Anderson, but I was also at Mass General. And I remember talking to a triple negative breast cancer patient there, and she had been in 5 clinical trials with all kinds of drugs. Wow. And it just overwhelmed me, the amount of chemotherapy that she had had over the years, and her willingness to try any new clinical trial that her doctor even remotely heard about to treat her disease. And so that's really what inspired me.
That's great. Wonderful background. Thank you for sharing. So Pam, who's up next? Dr. Gomillion, also from the University of Georgia. So Cheryl, let's give you a sound check and make sure we can see you.
Can you see me and hear me? Great.
Great.
So I guess I'll reverse mine, because my background kind of feeds into what we do now. So I first became interested in breast cancer research because of my mom, a breast cancer survivor. So I remember her going through the treatment, decision-making processes when I was in middle school. And so once I ended up at college as an engineering major at Clemson, so over in the other one of our enemies here, I actually was looking to get involved in undergraduate research. And I was in a different department, and I found this program, Biomedical or Bioengineering. And I talked to a professor there who told me about her work in the area of tissue engineering. And so it's exactly how it sounds, trying to engineer tissues of the body. And so she was working on projects to develop natural tissue replacements that could be used for breast reconstruction. And so this really struck a chord with me, having remembered my mom going through decision-making processes of, a prosthetic or reconstruction after having her surgery. And so when she mentioned that, she welcomed me into her lab, and then I ended up staying for graduate school and my doctorate in bioengineering there at Clemson. So I'm a triple tiger over here in Georgia. And so at that time, my doctorate work focused on, reconstruction options. So could we develop minimally invasive reconstruction options for breast cancer patients? And over time, the principle of tissue engineering is that we're mimicking something that would be naturally found in the body. And so over time, we started to get, really interested in, can we use these tissue mimics for something else? And so basically trying to answer questions about the disease itself. And so instead of, mimicking something that would be a filler for reconstruction, can we incorporate tumor cells into these model systems and create little miniature models that we can use in the lab to study how these cells are behaving? And so because there's a large proportion of adipose or fat tissue found in the breast, that was our target as a filler material for breast reconstruction. And so over time, my interest has evolved into trying to understand what the role of that fat tissue could be in the body and how it could affect tumor cell behavior. And so my lab now at UGA focuses on using these engineered tissue models that we can take into account physical properties of tissue, so softness or the stiffness of the tissue and how that can change over time as tumors form, but then also chemical properties of the tissue environment where fat tissue produces like proteins, hormones, and other factors that can affect tumor cell behavior. And so we're interested in combining these attributes into model systems that we can then use for studying cell metastasis or other processes associated with breast cancer.
Very good. Thank you. And now we'll go to Dr. Aneja. We'll see you in just a moment, Ritu. There you are. No, there's your picture.
Can you hear me?
I can hear you. There, welcome.
Okay, thank you. Thank you, Nancy. Thank you, Stephanie, for doing this. And thank you for all of the participants being here. So yeah, when I started studying cancer 15 years ago, I was looking at surrogates of intratumor heterogeneity, like centrosome amplification, centrosome clustering, cell cycle kinetics. That's what I was doing. That was during my postdoc work at Emory, and when I started my independent lab at Georgia State in 2008. And with an overall goal to glean prognostic information such as risk of metastasis, aggressiveness, and to identify druggable targets, of course. So at that time, I was very intrigued by the enormous disparity that I saw in breast cancer outcomes between Black women and white women, and particularly in the triple negative subtype. And that's the time when, and I can tell you, it was around 2012, that's when I started drifting towards racial disparity research, and my lab now at Georgia State focusing on studying the multidimensional complexity of racial disparity in breast cancer outcomes. So there are many, many questions that still remain unanswered. Why are tumors in triple negative patients, particularly those who are of African descent, they are inherently more aggressive than breast tumors in others. And I'm also very interested in exploring why there is a tremendous biogeographic disparity in triple negative across the world. And of course, the goal is to find biomarkers among TNBCs, triple negatives that can tell us which cancers will be aggressive, which will be indolent, which will respond to chemotherapy. So I'm using a transdisciplinary perspective because I don't think that you can answer these kind of questions just from a biological standpoint, and you have to bring all the different disciplines together. And as Mandi mentioned, triple negative is a very interesting subtype because it's kind of like it's a catch-all phrase that lumps together all the breast cancers that lack the ER, PR, and the HER2 receptors. So it is known for what it lacks rather than what it has. And so that's the interesting part. And so with support from Georgia Core and It's the Journey, in 2017, we were able to elevate the subject matter of triple negative to quadruple negative, because what we found was very surprising and interesting, that women of African descent, they tend to lose AR. They tend to lose androgen receptor. So we did a multi-institutional study, and that would not have been possible without the support, and which included 6 countries and 15 different institutions. And what we found that African patients, those who are from Nigeria, and some parts of Ghana. That was the 2 sites that we collected samples from. They are not 90% or more quadruple negative. They lack androgen receptor. And here also in the United States, we have women of African descent, they tend to be quadruple negative. So that is—
I'm going to ask you to pause there if you would, Dr. Aneja, because I want to make sure and announce to our our attendees,
yes,
that you can use the Q&A to ask questions of the researchers. And some of what you're talking about, Dr. Aneja, is what would be in the next question that I would ask. So please know that you can use the Q&A and the chat will be monitored, or the chat will be monitored, and then we'll field some questions to you. Can I go back around and kind of pick up on the brief introductions of the other panelists, and let's begin to move into this question about the depth and breadth of your research that Dr. Aneja was just referencing. So, Dr. Marcus, I want to go back to you. You're the person who's in a clinical setting where you have the opportunity to work with patients and doctor and physicians, medical oncologists more readily. I was interested in your overview of your work, where you saw the possible bridge to something that we're hearing about as a liquid biopsy. Can you give us just a little bit of background on that and how that might be a future application of the type of research that you're doing?
Yeah, absolutely. Great question. So the building I work at, Winsham Cancer Institute at Emory, is a combined research clinical building. And what that means is that basically the bottom 3 or 4 floors are clinical and the top 3 floors are research laboratories. And this really, and in reality does foster communication and joint research projects and just different opportunities that I know for myself personally, I wouldn't have been able to have. And that really has changed my research interests. I mean, we hear stories like we hear now. And, I walk past patients every day and, it changes your mindset on how you approach these questions, undoubtedly. So related to the question is on this idea of liquid biopsies, which is a diagnostic approach. So it allows for more or less non-invasive, so a blood draw, to determine if breast cancer cells and other cell types, other tumor types as well, are in the blood. And you can imagine that being in the blood is directly related to spread of the cancer. So the opportunity it provides is huge because it really allows for the first time to monitor in a repeated way, over time, if the tumor is spreading, and from a research perspective, what are those types of cells that are spreading? Because now it really provides that opportunity to change treatment over time, and to know earlier when it happens through the blood draw, and then to change treatment. And for us personally, it allows us to really look at those rare cancer cells that I was alluding to. And what is their genetic makeup? What is driving their spread? And again, ultimately, working with the clinicians, how we could then change our treatment to precisely target those cells in the liquid biopsy in circulation. And I'll stop there.
Very interesting. Excellent background and further edification about how research will be translated to diagnostic applications. So Dr. Murph, I'd like to go back to you. You mentioned a term that sounded like something out of science fiction when we talked, and that was delivering lethal cargo to cancer cells. Like, this is a pharmaceutical approach to attacking cancer. Can you describe just a little bit about that to our participants?
Yeah. Thanks, Nancy, for that question. So what we are working on is a particle, and think of the particle as like a tiny little bubble. So introducing thousands, perhaps millions of tiny little bubbles into a patient. And the bubble on the outside has, let's say, a GPS tracking unit. So it knows exactly where it's supposed to go. And inside the tiny little bubble is something that's going to be harmful to the cell, either eliminating the cell or resetting the cell back to a more normal function. This wouldn't necessarily be used alone. It would be more in a combination approach. However, the idea is that what's inside the tiny bubbles, you could think about it as lethal cargo, meaning there's some entity in there that would be lethal to the cell. But sometimes that facilitates resistance or even more cell killing. There are complications to doing that. Some of the newer drugs don't necessarily kill the cells. They just try to reset some of the bad functions that are going on inside the cell to make it more normal. And so you can use either approach depending on what you want to put inside these cells. But I really want to thank the Georgia Corps and It's the Journey for their funding, because thanks to the money that we received from our grant, we now have a patent that's pending on our tool and the innovation that we have been able to create and get working in the lab through that funding.
That's an outstanding accomplishment. So thank you, of course, for sharing that. Let me just move to Dr. Gomillion. Of course, I may think I understand more about adipose or breast tissue than some of the other areas that have been mentioned. So you talked about tell us a little bit more about how engineering aids research in metastasis regarding breast cancer tissue. I'm very interested in hearing you talk some more about that.
Sure, definitely. Thank you. So my background in engineering, and there are many researchers, across the country, the world, that are trying to apply engineering approaches towards like these same problems. One of the key things with engineering is just being able to implement different tools and developing different technologies that can help support this work. So for example, with the goal of tissue engineering, we're trying to make tissue mimics that can, tissues that mimic those that are found in the body. So in particular with breast tissue, in breast cancer cases, the tumor microenvironment plays a key role in how these cells behave and respond. And so using these tissue engineering approaches, we can develop models by combining cells and biomaterials and other chemical factors that will mimic the tumor microenvironment, and then that can be used to model theoretically the process of metastasis that's happening. So with some biomaterials, there are materials that will respond to cells that are producing certain proteins that might degrade or break down the ECM that we talk about that will allow tumor cells to migrate or metastasize out. And so we can mimic those processes in the lab. There are also other tools that like 3D printing or bioprinting that can allow for faster and more precise preparation of these tumor models and tissue models, so we can mimic and tailor those conditions. There's also engineering methods like microfluidics that will give you kind of a small scale, more high throughput approach at testing or evaluating these processes, but then also down the line that could be used for drug screening applications as well. So I hope that answers your question.
It does. It does. Very interesting. So Dr. Aneja, we'll come back to you. And you mentioned you were really rolling along there with your interest in global collaboration. This is something that we hear about more and more in Atlanta and in Georgia writ large. So can you tell us just a little bit about your global collaboration and why that holds promise in the study of triple negative breast cancer?
Yeah, thank you, Nancy. So yeah, in 2016, I founded ICART, which is the International Consortium for advancing research on triple negative breast cancer. And ICART today has about 35 institutions from 15 different countries and a whole 20 institutions only from Nigeria. So why I feel these global collaborations are important for triple negative, because of that, you know, biogeographic disparity all over the world. If you go to Africa, we have triple negative to the extent of 50 to 80%. And now the, you know, as I mentioned before, that the subject matter is now up to quadruple negative. So, and then coming to the US, we have only 10 to 15% if regardless of race, but if we factor race in, then we have about 25 to 40%. So it's important to get minimally admixed African samples to see race and associations. So one key objective of ICART is to obtain race annotated TNBC specimens from minimally admixed native African samples. So that is would allow us, because in the United States, the race is self-reported, and even African Americans, they have on an average 3 to 35% of European ancestry. Mm-hmm. So we need them to look at why these tumors are more aggressive in triple negative, why those tumors are more aggressive. We need minimally admixed samples as well. So that is one of the major reasons that we went global.
That's interesting, fascinating. So let me just make sure that I let all of you know that if you have a question, you can submit that to us and we'll forward it to our researchers. And it doesn't appear, unless I'm missing something, let me look here, that we have anything right now. So I want to just circle around. This will probably be a shorter question, but it's something that I'm really interested in that's a theme. So you all are, without a doubt, a multidisciplinary, highly qualified team of researchers. And I'd like to just hear a comment.
There are two questions.
Here, let me just make sure. I'd like to hear a comment about how collaboration impacts your research and your work. And why don't we just go back to Dr. Marcus, if you want to start with that, and then we'll follow with the other panelists, and I'll see if we have some questions.
Yeah.
There actually are two questions in the Q&A box. I don't know if you can see
Okay,
that.
why couldn't I see that? What you got, Dr. Gomillion?
Well, I'll ask it, yeah. So to Dr.
Okay.
Marquita Bass says, I'm an 8-year triple negative breast cancer survivor. Because TNBC is not one disease and the different subtypes have been proven to respond to different agents, why does the majority of research approach it as one disease? Layman's study 9 years ago has been reproduced, yet there still isn't a target treatment for early stage disease.
Yeah.
Who wants to take, good, Dr. Marcus?
Yeah, I think I could do part of it, but I'd love to hear also everyone else's thoughts. I think some of the people on the panel are more experts in triple negative than I am. So my feeling is this, that I think as a research community, we recognize this as more than one disease. I mean, we know that there are subtypes upon subtypes upon subtypes, and that in part is what makes this disease so complex, right? And I would add to it that also makes it challenging to treat. So I think the approach going forward, and I would acknowledge, I think researchers do recognize that these are probably multiple diseases. The approach going forward then, as is alluded to in the question, is that how then do we take this knowledge of multiple subtypes and then treat accordingly? I think the challenge is, just because we know that there are different subtypes does not necessarily mean that we know what the best treatment is for those subtypes. There could be things that are not targetable or druggable, as Dr. Aneja used the term previously. So I think that there are challenges in going from understanding the subtypes of triple negative to then treating based upon those subtypes. And I will sort of just mention the other question on collaboration. This is the example of why collaboration is important, is because we need to take our understanding of these research subtypes and collaborate with the clinicians to then translate that understanding into treatments. And I would emphasize that the only way to do that is through collaboration. And I buy into that for sure. And I'll let the other panelists, I saw the second question was related to, I'll let the other panelists chime in as well.
Please, who would like to go next?
Yeah, so I can add to what Adam, really said very eloquently, that yes, the challenge is intratumor heterogeneity, and that is what is failing therapy. And then triple negative, so many subtypes are known, and but they are all gene expression based, and it's not easy also to embrace gene expression profiling at this, in the current conditions, or wherever the clinic is right now. But yeah, and that's why my approach was to look at something immunohistochemical, and that was the androgen receptor, because a couple of years ago, there was a lot of buzz around using AR antagonists borrowed from the prostate cancer world for triple negative. But then there were a flurry of clinical trials that showed that even if you only 30% of women who were AR positive responded, and then the rest did not. So there was a lot which has to be understood in terms of the biology of triple negative. And yeah, so it is still a challenge, and the search is still on.
Absolutely. And so would others of our researchers, Dr. Gomillion, do you want to comment on this or the topic of collaboration?
I think yes. So there was one more question, but I can comment on the collaboration.
Oh, good. Okay, go to the question, please. I'm toggling back and forth.
No problem. So the second question, I guess, was kind of along some same lines with triple negative breast cancer. So to Dr. Murph, Marquita Bass asked, you mentioned that in the last 2 years, there have been treatments for triple negative breast cancer. As a patient who had early stage disease 8 years ago, the drugs are only for advanced and metastatic disease. What is the status for finding targeted treatments for early stage disease? Although most women survive, a lot of women deal with triple negative breast cancer recurrence within 2 to 3 years.
Okay, I'm happy to answer that. That's an excellent question. And a lot of the answer has to do with what treatments we have available, what approaches are used for breast cancer, and also how clinical trials are designed. So typically for early stage breast cancer, surgery plays a big role, radiation may play a big role, but drug therapy may not necessarily play a big role, albeit I do understand the question, and I do certainly agree with the question at hand. Typically, if you are going to run a clinical trial for a new drug, this new drug, you have evidence that it's going to work, but you don't know for sure what that's going to look like or whether that will ring true in, let's say, a phase 3 clinical trial. So when you enroll patients, we typically get less than 10% of all cancer patients enrolling in a clinical trial. And so with breast cancer, that's going to be a very small number of those breast cancer patients will have triple negative disease. And you also have to give informed consent and inform the patient, let's disclose the standard of care. You know, if you have early stage breast cancer, you have great treatment options with surgery, perhaps radiation, but with surgery, you have excellent outcomes for most types. And so as a patient, do you want to go and use the standard of care knowing that you may have an excellent outcome and benefit that? Or, you know, do you want to try a new untested approach where you're not really sure if it's going to work or not? And so you can see the more that we start to add on, that number shrinks. That's why typically when you see a new drug approval, it's for an advanced metastatic stage. Because at an advanced metastatic stage, the options become more limited and the outcomes become more limited. And so patients may be more apt to try and enroll in a clinical trial to see what might come out of that, knowing that the standard of care options, again, are more limited and so are the outcomes. And in addition, a lot of times when a new drug is approved, it starts out as an approval for an advanced stage or maybe third line treatment. because patients have gone through standard of care, and if those approaches haven't worked, then they're more open to trying a clinical trial. As we learn more about the drug, typically we can work that up into second-line therapy and even first-line therapy. And I'll give you a perfect example, atezolizumab, so the new immunotherapy. It used to be for, advanced stage lung cancer. It still has that indication, but now it's used as first line for small cell lung cancer. So again, as we learn more about these drugs, they can certainly be moved up into earlier stages once we have confidence in them and we can run those clinical trials with good data. So I'll now open it up to the panel to see if they have any other comments about that.
Anyone have comments? I know that one question we were talking about in preparing for this panel is, we know that COVID has impacted our work, the walk, the way that Georgia Core works, the opportunity to get together through this Zoom webinar, which is phenomenal, and it was available before, but now we're being very resourceful and finding out new ways to connect. I think one of the questions that we want to ask all of you as researchers, or each of you, is how has this pandemic affected your work? And so one thing that I think I would have a concern about is whether the resources going to COVID, might any of those be diverted from sources of research funding that you all have? Might this interrupt the trajectory of this extraordinary breast cancer research? So let's, ask each of you. And Dr. Murph, I'm looking at you, so maybe you want to be the first to just comment on this. And is there a COVID effect?
There is. I'm sure we can probably all touch on this. The government had our labs shut down, at least here at University of Georgia this spring. And we were slowly able to open back up in the summer. But if you were doing COVID research, you could continue on. So there were certainly incentives for, moving in that approach. But I'm sure this impacted everybody. They certainly didn't want undergraduates on campus doing research and graduate students. So, I'll ask the other panelists if they had the same experience that I did.
Dr. Aneja?
Yeah, as Mandi mentioned, yes, our labs were shut down, and we slowly came back, and then we had some cases of COVID in our labs, so we had to again re-shut down and quarantine our students, as well as get all the disinfection protocols followed for our labs and other places. So it's been really challenging.
Mm-hmm. Dr. Gomillion?
Yes, so I'm at the University of Georgia as well, and just exactly what they said. We had to shut down like March, and shortly after our spring break, students were not allowed on campus. Faculty, we could only come and do essential things, which was not related to our research. It wasn't considered essential at the time, even though it is for us, right? And so that was a big, just everything coming to a halt was really a big, roadblock for what we do. And getting started again, like it took some time. It's still taking time, to be honest, with students now considering, full return to campus, where before during the summer when we kind of had a slow approach back into, the lab, there weren't all the students here on campus, but now there's even more bodies present. So students are anxious and about it. Faculty and staff are also anxious about it. And so just trying to maintain some normalcy with the research lab has been interesting in this time of COVID, for sure.
And Dr. Marcus?
Yeah, so I'll echo what has been said. We had a very similar experience, and we put safety as our #1 priority. I think as we ramp up now, I think we and I'll maybe echo some of the other voices of leaders at Winship, is that we see cancer care and cancer research as essential, and we are practicing the right safety measures. We have strict policies in place. It's a challenge. But we're operating in a safe way. And, we're going to try our best to continue to do so. I kind of remain optimistic that we will have the same level of funding for cancer research going forward. I think this is critical. I think that we're, across many cancer types, we're making progress. I mean, we're at a really important point, and I just want to see that continue. I know I'm excited by the research I hear from everyone else and our lab's research. So I feel like we have a tremendous upward trajectory, and we want to maintain that.
So I'm going to go around one more time and ask each of you, because in keeping with our theme, highlights and hope, what you're hopeful about. So Dr. Marcus, I'm looking at you right now. Can you just make a comment or two about what you're hopeful about in this domain of breast cancer research and what keeps you going?
Yeah, so I would say, for me, just the passion and curiosity to make a difference is absolutely critical. That keeps me going for breast cancer research. As I mentioned before, walking every day into that building now with a mask on, is quite motivational. I mean, I think that, like many things, research is difficult, it's complex, but I think keep pushing forward. Perseverance is key, on many levels. And, I'm excited. I hear all the research that goes on here. I mean, it's so exciting and it's so multidisciplinary and it's getting extremely collaborative, and all those things are just, empowering. And we hope to really translate that to the general public, to our patients. So it's exciting times, I'll say.
Very hopeful message. Dr. Gomillion, do you have a word of hope for us and our participants?
Yes, I think similar to what Dr. Marcus said, just, staying, it does get challenging, right? This research is up and down, many hills and valleys that are here in the process. But I think what keeps us going is just, the long-term thought about how we can really impact patients' lives. For me, in this, basic research lab and educating students, right, in the classroom, and the students that come through the lab, they are very interested in how we can make a difference. It's sometimes hard for them to understand that there's an iterative process here and that it's not going to happen overnight. But I think that's what keeps me going is, their enthusiasm when they come in the door and how, excited they are to make a change and continue to drive the hope for others. And so just thinking about, long-term, what we can do to help people, that's what really drives us here.
Dr. Aneja, message of hope. Oh, can't hear, unmute.
Yeah, so I completely agree with what all has been said. And I feel that partnership is very powerful, and coming together and cutting across disciplines, bringing basic researchers, translational researchers, epidemiologists, public health experts, biostatisticians, social scientists, and bioinformaticians, statisticians, everybody under one roof. That's when I feel that magic can happen. And that's what keeps me going.
That's great. Dr. Murph?
So what gives me hope? That survivorship is excellent. There's always improvements that we can make, but that patients now have more options than ever before. If they want to do surgery, what kind of surgery, reconstruction, radiation, drug options, and then new drugs like Trodelvy, where now you can target what we thought was untargetable. That gives me a lot of hope for the future. And every time I have a friend or a colleague that moves from patient into survivorship, that always gives me just overwhelming hope in terms of, look how far we've come from decades ago to today.
Mm-hmm. Outstanding. You all are such a dynamic team of Georgia researchers. We're so very proud to have you here and to be able to support you. And I also want to say that there are other researchers who have received funding. And if you participants like this webinar in this format, then reach out to It's the Journey or to Georgia Core and let us know, and we can do more of this. So that would be terrific. And we want to encourage and support your collaboration. The more successful that we are, we know, we can see and feel the great work that you're doing and how this will be extraordinary for the future of breast cancer research. So I didn't mention, Stephanie, that we have had another partner too, A Cure in Our Lifetime, and we provided a 3-way grant this last year. So in our next webinar, you'll be hearing more about that. We'll let you know. But thank you all. And Stephanie, I'm going to turn it back to you. I have a feeling you have a rah-rah close for us.
Well, I mean, I did hear Dr. Marcus say walking into that building every day. So I just want to remind everybody that if you haven't signed up for the walk, the 2-day walk is now virtual. It's an experience, and we're having a lot of fun on this virtual app. And the actual walk will take place September 29th through October 3rd, and then we're going to do a closing. And, you know, the walkers and our sponsors, those are the ones that are doing all of the work. They're walking their miles. I think in the last 2 weeks, we walked over 9 million steps. So, Dr. Marcus, I don't know if you want to try to match us step for step, you know, walking into your building. I'd be happy to watch that happen. But you know, we love you guys, and we love kind of having this experience and seeing where those dollars are going. And I know that the walkers are so interested, and they're going to want to hear more about, you know, how COVID has affected that and the struggles that you guys are having. But they want to know, I mean, this is great stewardship of their dollars. They're walking those 30 miles and they're wanting to support you guys. And so, you know, the more you can be engaged with them and the more they can be engaged with you, I think it's going to be awesome. So we appreciate you guys. Hopefully all the questions were answered. And I do believe this was recorded, so we'll hook it up to our Facebook, and so people can see that as well. But we thank you guys very much. And of course, we thank our walkers and our sponsors and all of that. So it's awesome.
Very good. Well, thank you, our partners at It's the Journey, Stephanie and Pam. Thank you to the Georgia Core supporting cast who do a great job in every way, Katrina Mitchell Davis and Rebecca Wallace. And we look forward to doing more together, and we hope to engage more directly with all of you as researchers in our efforts to raise money and support your work. So thank you all. We appreciate it very much. Let us hear your comments.
Yay.
Thank you.
Bye-bye.